Good afternoon and welcome to the PRISM e-Symposium. I am Dr. Gabriela Sanguinetti and I will be serving as your moderator for this session. Oscar Cifuentes is my co-moderator.
Hi everyone, good luck.
You're on the Biomedical Sciences concurrent session. This session will go from 5:35 to 6 p.m. In this session, we will hear from students presenting on topics related to gene expression in diseases like rheumatoid arthritis and Parkinson's disease. If you have questions, please use the Q&A button below to write them and send them in. We will select questions at the end of each presentation. Next slide, please. Our first presenter in the second session is Nickolas Almodovar, and the title of their presentation is Relevant Exosomal microRNA Identification in Rheumatoid Arthritis. Next slide.
Thank you, Dr. Sanguinetti. Rheumatoid arthritis affects more than 1.5 million Americans every year. And patients of rheumatoid arthritis suffer from bone erosions, joint deformities, inflammation, lack of mobility. An example of a patient's hand is shown on the right. Eventually, this leads to further complications such as heart disease, potential for diabetes, or lung disorders as well. So to better understand, it's important to better understand this disease in order to develop novel treatments, interventions, and therapies for this disease, which causes a lot of adverse effects. Next slide, please. Currently, scientists see that there most likely is a genetic and environmental factor influencing the underlying biology or pathogenesis of rheumatoid arthritis. We can see that interaction through the lens of epigenetics. MicroRNAs are an example of epigenetic regulation. On the right, you can see a set of maturation steps, which leads to an active microRNA molecule. These active microRNA molecules can regulate the function inside of the cell. Genes in the cell are expressed in the form of messenger RNAs. MicroRNAs can bind to these messenger RNAs, tagging them for degradation or preventing their transcription or expression of to a newly formed protein. One single type of microRNA can regulate many different messenger RNAs at the same time. Next slide, please. MicroRNAs can travel through the body. In the image, you see microRNAs in pink packaged into these little bubbles called exosomes. These exosomes can be transported through the body to a target cell, such as an immune cell. When they bind to this immune cell, or when they reach this immune cell rather, they can regulate gene expression inside that target immune cell. These exosomes serve as a way of protecting microRNAs as they travel. Scientists have seen some association with microRNAs in general with rheumatoid arthritis, but we still haven't explored sufficiently exosomal microRNAs and their associations with rheumatoid arthritis. Next slide, please. Thank you. And that's where my project comes in. The goal of this research project is to identify exosomal microRNAs associated with rheumatoid arthritis. And for this project, 27 patients with RA, rheumatoid arthritis, and 18 healthy controls, patients without, or people without rheumatoid arthritis, were recruited for this study. From them, we collected serum samples. Next slide, please. In the serum, we were able to extract exosomes. We then purified the microRNAs found inside of those exosomes. We were able to then quantify 800 different microRNAs using N-counter quantification and perform data analysis using N-solver. We then applied a stringent criteria to select the most relevant microRNAs and perform statistical analysis. Next slide, please. When we performed N-solver analysis, we were able to identify 72 exosomal microRNAs differentially expressed in rheumatoid arthritis. We show this data here on a heat map. The point we tried to cluster this to see which microRNAs are most relevant in select populations. So down the left, you see a list of microRNAs, all of those that we found relevant. Along the top, we have every sample listed, denoting either a healthy control, person without RA, or an RA patient. In blue and green, you can see relative expression, green being a higher level of expression of select microRNA and blue being a lower expression. The red box indicates a clustering of samples. Next slide, please. After applying stringent criteria to identify the most relevant microRNAs for our study, we were able to narrow down our list to 13 microRNAs. You can now see a little bit clearer the list of microRNAs we were able to identify down the left and all the samples we used in the study along the top. Here, once again, we performed an unsupervised hierarchical cluster, and we see that for the most part, there's a clustering of rheumatoid arthritis patients, as shown in the red box. A lot of these patients have a higher expression of those select microRNAs based off of those rows. Thank you. Next slide, please. Here we show the levels of each of those exosomal microRNAs differentially expressed in patients with and without RA. As you can see that they're from the 13 we identified, 4 were identified in the literature previously. On each representative column shows either levels in patients with or without RA respectively, and a p-value showing how significant this change in expression is. Thank you. Next slide, please. So overall, in sum, we were able to identify 72 microRNAs that had different levels, and then narrow it down to 13 microRNAs applying stringent criteria. From these 13 microRNAs, we identified 9 that were newly identified in the study, whereas 4 had been previously identified in the literature in at least 2 or more papers. Newly identified microRNAs have the potential for strengthening clinical diagnostic, follow-up, and response to treatment in rheumatoid arthritis patients. In the future, we would like to validate our work through the use of qPCR, as well as investigate potential correlations between the levels of these microRNAs and clinical characteristics. Next slide, please. I would like to thank my mentors, Dr. Delgado-Cruzata and Rodriguez Alvarez, for their guidance and support through this project, as well as Tonya and Bravo for her technical support with nanostring analysis. I'll also like to thank the clinicians who helped collect and sample and process these samples, as well as my funding sources, including PRISM and the Macaulay Honors Program. Thank you.
Thank you, Nick. As a reminder to the audience, if you have a question, please use the Q&A box, click on the Q&A button and submit your question. So I have a question, Nick. You mentioned that out of the 13 microRNAs that you found, 4 of them have been previously identified, correct?
Yes.
Out of those 4 microRNAs, have any of them been implicated in other inflammatory diseases? Any other inflammatory diseases?
These microRNAs in particular have been somehow associated with rheumatoid arthritis in particular. They may have some association with other inflammatory diseases that I do not know of as of now.
Thank you. Next slide. Our next and final presentation is a joint presentation with Joy Alejanzalez and Samara Pettie. And the title of their presentation is Epigenetic Modification by Mancozeb and Astrocytes in PC12 cells using JMJD3. Next slide.
Hi, my name is Joy, and I will be presenting the first half of our presentation. Can you all hear me?
Yes.
Okay. Can you all see me? I'm not entirely sure.
Yes.
Our research makes an impact in the world by shedding light on the dangers and harmful effects of using pesticides and fertilizers. As the population grows, the need for food continues to climb, requiring us to use fertilizers and pesticides to meet the demand. In the photo shown on the left, a farmer uses a backpack sprayer to apply pesticides to his rice paddy field. Notice how he's covering his face with a cloth using the pesticides. That is due to the higher risk and exposure dealing with these fertilizers and pesticides daily. Other groups also at risk of increased pesticide and fertilizer exposure are people who handle large quantities of produce like grocery store workers and produce distribution plants. Next slide, please. Mancozeb is a dithiocarbamate containing essential metals, manganese and zinc, which are commonly used in fungicides. Manganese is an essential element for supporting development, growth, and neuronal function. Next slide, please. Mancozeb has low reported toxicity in mammals, which is shown in studies that relate Mancozeb to hypothyroxemia and other adverse effects on adult thyroid hormone systems. In humans, hypothyroxemia early in pregnancy is associated with adverse effects on the developing nervous system and can lead to impaired cognitive function and motor development in children. Additionally, overexposure to manganese can lead to manganism. The display of Parkinsonian-like symptoms, Mancozeb can potentiate Parkinsonian toxicant MPP plus toxicity at non-toxic doses. Next slide, please. The problem we are trying to solve is, why does Mancozeb exhibit potentiation effects at non-toxic levels? Our hypothesis is that increasing the concentration of Mancozeb in astrocytes and PC12 cells will yield increasing expression of protein JMJD3. Our aim is to investigate the effect of Mancozeb on expression of epigenetic modulator JMJD3 in astrocytes and PC12 cells. Next slide, please. Let's start off by understanding what is epigenetics. Epigenetics is the study of the alteration of genes phenotypically without creating outstanding changes to the DNA itself. There are two main types of epigenetic modificators: DNA methylation and histone modification. In DNA methylation, enzymes called DNA methyltransferases add methyl groups to DNA on the 5' position of cytosine nucleotides that are found next to a guanine nucleotide in a DNA sequence. There are also multiple types of histone modifications, which are catalyzed by a number of enzyme families. Some of the categorized modifications include acetylation and methylation. There are also multiple types of histone modifications, which are catalyzed by acetylation and methylation of histones H3 and H4. JMJD3, which is short for Jumanji domain containing protein D3, is a demethylase that removes di and trimethyl groups from lysine 27 on histone 3. Next slide, please.
Ooh, so sorry, I forgot to unmute myself, guys. For the cell cultures, we used astrocytes and PC12 cells and incubated them at 37 degrees Celsius and a 5% CO2 incubator to stimulate human body conditions. They were chemically treated for 24 hours with Mancozeb and manganese chloride in the respective doses of 5, 25, and 50 micromolars. Then we performed a nuclear protein extraction of the control and treated cells. And lastly, we did a JMJD3 ELISA assay. Next slide. Next slide. Okay. With the ELISA assay, we coated the wells with antibodies. Then we applied an antibody detection. The JMJD3 binds to the antibodies, and then we applied an enzyme substrate, and the signal was measured by a plate reader. The higher the signal, the higher the JMJD3 expression. Next. In the PC12 cells, we can see that there was no significant expression of JMJD3. While in the astrocyte cells, we see that JMJD3 is expressed only at 50 micromolars of Mancozeb, while the expression of JMJD3 increases at a dose-dependent manner with the manganese chloride. Next slide. In conclusion, we found that astrocyte cells were more sensitive to manganese chloride triggered JMJD3 activation, as the JMJD3 signal increased at a dose-dependent manner, while in PC12 cells, both the manganese chloride and the Mancozeb showed no effect on JMJD3. Danieli showed that neurons are susceptible to zinc toxicity as compared with astrocytes. However, there's a higher rate of mitochondrial dysfunction in astrocytes than in neurons triggered by zinc. And Merkwink suggested that JMJD3 is a positive regulator of lifespan in response to mitochondrial dysfunction across the species. Next slide. In the future, we would like to uncover the relationship of Mancozeb and manganese with other epigenetic related proteins such as HDAC1 and DNMT1, evaluating the methylation status of histones and/or by determining if antioxidants like curcumin can reverse the epigenetic changes triggered by Mancozeb. Next slide. We would like to thank Dr. Cheng, our mentor and advisor, as well as our friends and family, and PRISM for giving us the opportunity to present our research to everybody today. Thank you very much for your time.
Thank you, Samara and Joy. I have three questions from the audience, so I'm going to indulge all of them. The first question is, why did you use astrocytes in PC12 cells?
Well, we used astrocytes and PC12 cells because, well, I'll answer the astrocyte ones first. We use astrocyte cells because glia cells are found in the CNS and they're housekeeping cells, and they maintain the CNS environment. And we chose PC12 cells because they're easy to maintain and easy to manipulate and to introduce extra genes inside the cell.
Thank you. I have another question. The question is, where are the astrocytes from? Do you think that astrocytes outside of an in vivo setting have the same epigenetic characteristics as those in the brain that are interacting with many other cells in the brain? So do you think your results are translate to in vivo or represent what's happening in vivo?
Well, astrocytes are the most abundant cells in the brain. And to answer the in vivo versus in vitro, it should basically function the same way. Our research approach stimulated conditions in the human body. So to replicate this experiment in vivo, we would essentially not have much of a difference in terms of how we conducted our research approach.
Thank you. And the final question is, why is there a different response between astrocytes and PC12 cells in JMJD3?
PC12 cells have been linked to transiently express JMJD3 levels due to the NF-kappa B light chain pathway, which is a pathway that promotes the expression of these proteins. So we think that that is the reason why it is a different expression than in astrocytes.
Great, and I have one last question. Could you have looked at another protein instead of JMJD3?
Yes, our lab looked at HDAC1 and DNMT1. HDAC1 is a histone deacetylator, and DNMT1 is a DNA methylator.
Great. Thank you so much. And with that, we conclude our session. I would like to thank our presenters for their work and ask everyone in our audience to send them a big round of applause. We will begin now in 5 minutes with our next plenary session, in which our keynote and PRISM alumna, Dr. Olivia Orta, will discuss her career trajectory from John Jay College to Harvard University, while sharing lessons learned along the way. To join the plenary session, visit www.jjay.cuny.edu/PRISM and follow the link at the bottom. Thank you.